Alright, this video is AP Bio Review, Unit 1. I know you guys said that Unit 1 wasn't necessary. You didn't want me to review it. But after looking over it, I feel like that was a necessary thing. Just a few things here that I feel like you may miss, or something that we talked about. This is some of the basic chemistry ideas. Um, One thing, you need to make sure you understand the difference between the different kinds of bonds. Whereas covalent bonds represents shared electrons, ionic bonds represent transferred electrons. And this is with molecules that form ions like sodium and fluorine. They're going to be, and what are they going to be doing? They're basically going to be transferring this electron back and forth between, and they form an ionic bond, which is like a kind of like a crystallization. Just like get salt here. And then hydrogen bond, and this has to do with polar molecules. All right, and so there was some confusion about this as well, I know, at one point. And so usually when we talk about these, we're talking about um, water molecules. So here's a water molecule, and so a water molecule can form several hydrogen bonds with other water molecules. Now, how does that work? Well, a water molecule is polar because the oxygen end of the atom, because it has such a high positive charge, all those, I mean, it's 8 protons, most of the electrons are going to be around the top. So this end is going to be slightly negative. Well, that means that this end is going to be slightly positive. Well, this other water molecule is going to be attracted to that slightly positive end on its slightly negative end. And that bond right here is a hydrogen bond. It's a weak bond, but it is probably the most important bond in biological systems. And so make sure that you understand the difference between those three types of bonds. We did talk about van der Waals forces. These are also temporary bonds, and these are really big molecules when all the electrons tend to move to one side of the one side of the molecule, and that leaving one side positive and the other side negative, and that's very temporary. All right, a few other things. We talked about some of the properties of water, like cohesion, adhesion. Um, Not too difficult. Cohesion, water likes itself. Adhesion, water likes other things. Talked hydrophilic, hydrophobic, and again, words you guys know. Um, we also mentioned the functional groups. It may be a beneficial thing for you to spend some time looking at those. I can just draw them up here pretty quick here. The hydroxyl group, the carbonyl group. This is where you have a carbon and one double bonded oxygen, and it could be either an aldehyde or a ketone. And a ketone is when it is located um, in the middle of the molecule, like this. And an aldehyde is when it's located on the end of the molecule. You know, where you have like this. That's an aldehyde. So aldehyde, ketone, two types of carbonyls. You have a carboxyl group, and this is usually this will do with an acid because it readily gets rid of hydrogen. Um, it's very similar to the carbonyl. but it'll have that hydroxyl on it, and it'll readily get rid of that OH and readily get rid of that H and have a charge be negative. Amine, this is where you have a molecule that looks like that, oftentimes has a positive charge. Sulfhydryl, and phosphate groups, and these are all groups that you should be familiar with because you never know when one of those fun molecules might rear its head on the test. All right, four functional groups of macromolecules. Four groups of macromolecules, and you have carbs, carbohydrates, lipids, um, proteins, and nucleic acids. All right, and each one of these has particular properties. We spend some time on each one of them, some more than others. So, carbohydrates, this is molecules that contain carbon, hydrogen, and oxygen, and usually in a particular order. If your carbon, well, it's just like I had it, and it's in a 1:2 to 1 ratio. So something like glucose, for instance, 6:12:6, always going to have, or most of the time going to have that ratio. There's a couple times where you can have more than one sugar, where it has more than one. But you have monosaccharides, which is one piece, and this would be like a glucose. You can have a disaccharide, which is a two-piece, like this is one of those examples, for instance. This is sucrose. And then you can have a polysaccharide. This is where you just have more than two. And some important carbohydrates to remember: starch. This is how a plant store energy. So starch is plants. Easy to remember that because of potatoes. Glycogen is animals. No really easy way to remember that one. Cellulose. This is plant cell walls. And chitin. This is like exoskeletons and fungal cell walls as well. Nothing else on carbohydrates. Their purpose is they serve as the primary source of energy for all organisms. This is the source of energy that most organisms will break down readily. Next is lipids. And lipids, real quick, carbohydrates, the monomers, monosaccharide, the polymers, polysaccharide, obviously, and the bond that joins between, it's one of those fun words, the bond that joins between two glucose molecules is called a glycosidic linkage. All right, it's a covalent bond. With lipids, primary function of lipids is to store energy. Lipids most lipids are hydrophobic. That means that they are afraid of water. They're composed of hydrocarbons, meaning that they're mostly made up of carbon and hydrogen. Two main parts. They have a fatty acid, and this is this long chain where you have the carbons and the hydrogens. There is a glycerol molecule, and this is what holds these fatty acids together. Glycerol molecule. And so you have something that looks like this, where you have the fatty acid chains. And you have some molecules that have, whereas this is the glycerol. Some of these molecules have 3 fatty acid chains, and they're called, of course, triglycerides. And the linkage that they form here is called an ester linkage, and it's a covalent bond. There's triglycerides. This is kind of your basic fat or oil. We talked about the different kinds of triglycerides. Sometimes you have a saturated fat, whereas every single hydrogen spot is completely full, that no carbon can pick up another hydrogen because there are every hydrogen. There's just no spots for them. All right. Whereas an unsaturated fat might have something that looks like this, where you have a bent, where there's a double bond, and so there's still room for more hydrogen. And you can have a monounsaturated fat where it only has one double bond, or you could have a polyunsaturated fat that has more than one double bond. Hydrogenated fats, or trans fats, these are the dangerous ones. These are the ones where they purposefully put hydrogen into them, and it makes them solid at room temperature, like margarine is an example of this. And another important lipid, obviously one that we've talked a lot about, is something called a phospholipid. And this is where you have this hydrophilic head with two fatty acid chains. And these fatty acid chains are hydrophobic, and these form your cell membrane, because you have them turning in like this. And each one of these phospholipids form part of your cell membrane. Next is proteins. Proteins. Another name for proteins, polypeptides. Proteins contain, or they're made up of amino acids. An amino acid has a particular look to it. The R group is different in every single amino acid. The R group in one of them is just an H, whereas the R group in some of the other ones are these long, intricate chains. There are 21 natural occurring amino acids, or 20 naturally occurring amino acids, and all of them have this different R group on them. Amino acids will join together with a peptide bond. And one thing that I haven't mentioned yet, anytime you have these molecules joining together, these macromolecules join together, it is called a dehydration synthesis. And that's where water is pulled out of the molecule. Water is pulled out of the molecule, and they're joined together. So you have, see if I can just kind of draw a quick amino acid here. And then the other side, you're going to have the amine group of another one. Actually, the amine group of this other one is going to hook up with it. And what happens is everything erases and gets undone. What happens is this OH here, and one of these H's here will break off and become an H2O, and then these two will join together a little more pretty than that. But so basically the H2O is taken out, dehydration, synthesis, those two pieces are put together. Now the opposite of that, it's called a hydrolysis. hydro meaning water, lysis meaning break. And this is where water is basically used to break these two things apart. And you get back to where you were at the beginning. So hydrolysis and dehydration synthesis, two very important concepts in micromolecules. Now back to proteins. Proteins have, again, they're formed by these peptide bonds, and proteins will make, for their primary structure, will make these little chains, whereas each one of these little balls is an amino acid. And this is just a string of this is just a string of peptide bonds, so you have a polypeptide. Now, that string, certain parts of it don't like each other and certain parts of it do, and so eventually they'll start to curl up. And so for the secondary structure, what you can have is those chains can form a sheet, which is called a beta sheet or beta pleated sheet, or can form an alpha helix, which is kind of where it looks like this. And normally what happens in any given protein, you'll have the secondary structure will form several of these pleated sheets and alpha helixes, and the bonds that are between here are hydrogen bonds, so fairly weak bonds. And the peptide bonds are covalent bonds. Didn't mention that. So hydrogen bonds. But when you get to the tertiary structure, many proteins are done at this third stage. But the tertiary structure is where you can have multiple secondary structures joining together here to make some kind of shape that looks probably nothing like that. But the way that those multiple tertiary structures together join together with the sulfurs of some of the amino acids, a couple of the amino acids have sulfur in them. Cysteine is one of them. And the cysteines will kind of touch one another and they form what's called a disulfide bridge. Now these are very important. These are covalent bonds, very strong bonds, and these are the bonds that hold the protein together. Now sometimes you can have a protein, and oftentimes you can have a protein that has a quaternary structure, and essentially what a quaternary structure is, that's where you have multiple of these tertiary structures coming together, kind of fitting together in different ways. And that works together to do some particular job. They're kind of woven together. A hemoglobin molecule actually has 4 of these, and right smack dab in the middle of it looks like a nice iron color. Is this big old iron molecule, and that's what binds oxygen, and that's what allows your blood to carry oxygen. So those are the 4 structures of proteins. And to denature a protein is to systematically break those bonds. You can do that with temperature, just simply heating it up. You can do that with, that's what an egg does. You're basically denaturing all the proteins in the egg. You can do that with pH. Lower the pH or really high pH, you can break all proteins. Or you can do that with salinity, high amounts of salt. Now, our last, but certainly not least, is add in nucleic acids. And nucleic acids, these are your things like our friends DNA and RNA. Now, they are made up of individual units called nucleotides, and a nucleotide is made up of 3 pieces, and we'll try to do this here. You have a 5-carbon sugar and this is where the molecule gets its name. It's either going to be ribose for RNA or deoxyribose for DNA. Then you have a nitrogenous base, and this nitrogenous base can be either one ring or two rings. The ones that have one ring are called purines. This is adenine and guanine, and the ones that have two rings are pyrimidines, cysteine, thymine, and then in the case of RNA, uracil. Now, I believe we got this backwards. And then here on the end is a phosphate group, and it has a little bit better picture than that, but you get the idea. There. So a little bit, I fixed my drawing, as you can tell. That's a better picture than what I gave you. And so you put multiple nucleotides together, then you make a nucleic acid, DNA, RNA. And these can be single chain, like RNA. And so RNA has a single chain. It kind of forms this helix structure. And then DNA is a double helix. Now, the bonds that are between the two strands of DNA are hydrogen bonds between this base and its corresponding base. Those are hydrogen bonds. However, the bonds that form between the phosphate of one molecule and the sugar of the next molecule are called phosphodiester bonds. Phosphodiester bonds. Now, nucleic acids, again, very important, make up DNA. We talk a lot about this in the genetics unit. Now let's look a little bit at, also talk about membrane structure, transport molecules, that sort of thing. We talked about the different kinds of transport that happens across the membrane. You have integral proteins that run all the way through the membrane. You have peripheral proteins that don't. These are things like your G protein complexes that we talked about in the cell signaling unit. Cell components, I feel like you guys are pretty good there. We don't need to necessarily run through a lot of that. I do want to talk about diffusion. Diffusion is simply particles moving from an area of high concentration to an area of low concentration until they are at equilibrium. And so they will move until they're equal on both sides and there's no more movement. That means that there's also no energy to be used. Nothing can move, nothing will move. Now you can build up a concentration gradient, but that requires energy to do that. But if you do that, then you have energy to be released and to do work. And this is how a lot of the processes in your cell work, is just simply by using these concentration gradients and then letting go of them in this kind of cycle. Now we did a couple of math. We did one type of math problem with this where we did total water potential. And I don't suspect that this is going to be a difficult thing for you. The only thing that was a little bit difficult was for some, was this idea of the solute potential. And the solute potential is always negative. Then you have some sort of ionization constant, a concentration R, which is a given. It's 0.083, and so that'll always be given to you. and then T, which is always in Kelvin. Now, the ionization constant has to do with the number of ion types present. And so if you are dealing with something like sodium chloride, for instance, and that is your solute, well, there are two ions in sodium chloride, and so the ion constant is going to be 2 because you have two ions, sodium and chlorine. Whereas if you have something like another popular one that you might see is sucrose, what I mentioned earlier. All right, maybe perhaps the third time I can write this. No. Now, this has no ionization constant. It's not going to ionize in solution readily, and so the ionization constant will be 1. And so usually going to be 1 or 2. I can't imagine that it would be that too complicated there. The pressure potential, psi p, is going to be negative or positive depending on where the pressure is coming from. A pushing pressure, I've shown you systems like this where you have some sort of plunger or something, and if the arrow's going that way, it's pushing. That is a positive pressure, whereas this would be like a negative pressure, like pulling a plunger. And each side of a system is going to, oftentimes you see this U-shaped thing here, and you have a little membrane across here, and maybe there's a plunger here, and the water's like real low there, and psi p on this side is going to equal 0, whereas psi p on this side, because it is a positive pressure, is going to equal 2. Well, the solute potential, let's just make one up here. On this side, it's going to be 0. Where on this side, there is sodium chloride. And so we have psi s equals negative I CRT. And we'll just say that it's ionization constant because it's sodium chloride is going to be 2, so negative 2. The concentration is going to be 1, and you have 0.083, and the temperature is going to be a balmy 37 degrees. And so we add 37 to 27 degrees, excuse me, so we can have that nice 300. So 27 plus 273 is where we got that 300. And you just do the math here on my trusty calculator to and you get a 49.8 negative. This is negative 49.8. That's a lot of salt. And so where's this going to move? Well, the water moves from a high total potential to a low, and so water's going to move from high to low. This is the total here. Just add these up. The total is going to be negative 47.8, and this one's going to be 0. And so which way is the water going to move? This way, from high to low. Even though this is pressing down, the water's still going to push against it because the water wants to move where there's a lot more salt. It's wanting to equal it out, and so the ending up system might look something like this, and the plunger might get pushed back up against its will. Something along those lines. You kind of get the idea of that problem. Those problems aren't too difficult. Again, mitochondria, chloroplasts, all those kinds of things, pretty easy. I feel like you guys are pretty strong on that. I will make a video for you on photosynthesis and cellular respiration, but for now, this is a good biochemistry review.